Hello, my name is Abigail, and I'm talking about antioxidant activity in avocado and banana peels, comparison of extraction method. Avocado and banana peels differ in their fruit matrix, and this this difference influences the type and availability of bioactive compounds that are within their tissues, particularly in their peels. These peels often contain antioxidant com These pills often contain phenolic compounds with antioxidant properties, and these antioxidants help to neutralize reactive oxygen species. And these reactive oxygen species are unstable molecules, and when they are in excess, they cause oxidative stress. However, these compounds have to first be extracted, and the efficiency of recovery depends on the extraction method, which depend on the temperature, the solvent type, and the level of mechanical disruption. I used 4 different extraction for these pills. I used the steeping, the maceration, the ultrasound assisted extraction, and the Soxhlet. My hypothesis was that the Soxhlet extraction will yield the highest antioxidant content from avocado and banana peels compared to steeping, maceration, and UAE. The objective was to compare the extraction methods using antioxidant assay and identify key phenolic compounds contributing to antioxidant activity using the UPLC. For the metals, the peels were washed, oven dried, using the oven dryer, as we can see here, and the samples were subjected to, and they were ground with the mortar and pestle, and for each fruit sample, we had 3 different individual independent biological replicates of each fruit, and after grinding, they were subjected to steeping, maceration, UA, and Soxhlet, and the extracts were then filtered, concentrated in a centrifuge. Supernatant were then collected and diluted and prepared for the spectrophotometric assay and the UPLC, chromatographic assay. For the spectrophotometric assay, we're testing 2 different assays, the FNC and DPPH. For the FNC, we're trying to determine the total phenolic content within each pills from the extraction, and as we can see here, we found that steeping had lower significant difference from maceration, ultrasound, and Soxhlet, and steeping again in banana had a lower significant difference from maceration, ultrasound, and Soxhlet, and maceration also has significant difference between Soxhlet and UAE, but there was no significant difference between Soxhlet and UAE for banana, and for avocado there was no significant difference between the maceration, the UAE, and the Soxhlet. For DPPH, we see that steeping and ultrasound had no significant difference, but there was another difference between the maceration and the Soxhlet and the maceration and the Soxhlet. And for the banana, we could see a very interesting trend here. Steeping also came out to have a lower significant difference from the others, but maceration had no significant difference from UAE and Soxhlet, but ultrasound had significant difference from Soxhlet. And for the chromatographic image here, we can see that we're able to identify and quantify the phenolic compound which we tested for vanillic acid, and the avocado had a very high peak intensity here with a high concentration from the avocado Soxhlet acquired. The result varied based on each extraction method, on how each extraction method interacts with the plant matrix and solvent type. And we can see that the methanol-based extraction showed higher phenolic extract than the water-based, because steeping was done using water and maceration, ultrasound, and Soxhlet were done using the methanol solvent. And the Soxhlet had produced high phenolic recovery through continuous heating and solvent reflux, while UAE had showed strong antioxidant activity, as you can see, not like the highest, but like there was prominent due to ultrasonicated cell disruption, and maceration and steeping relied on passive diffusion. Peak intensity is higher in avocado succulent, supporting the spectrometric assay here, showing that findings indicate greater phenolic extraction. Future studies should focus on further optimizing extraction conditions and exploring combined extraction techniques to improve both yield and preservation of antioxidant compounds. And these are the references I used. I would like to acknowledge my lecturers and my professors for supporting me all throughout the experimental guidelines. Thank you.